Hello students, we are doing previous year's PMT questions based on NCERT core content. In this video, we will discuss 30 MCQs from chapter 6 Molecular Basis of Inheritance. Question number 1. Which of the following statements is correct? Out of 4 given statements, statement A is correct, that is adenine pairs with thymine through 2 hydrogen bonds. Adenine thymine से पेयर करता है by 2 hydrogen bonds and guanine cytosine से पेयर करता है by 3 hydrogen bonds in DNA molecule. So mark A as a correct answer. Question number 2. Purine found both in DNA and RNA are. DNA and RNA both have purine and pyrimidine bases. Purine bases are adenine and guanine, which are same in DNA and RNA. So correct answer is C, adenine and guanine. Mark C. Question number 3. The association of histone H1 with nucleosome indicates that histones are positively charged basic proteins which help in packaging of DNA, and the association of histone H1 with nucleosome indicates that DNA remain in its condensed form. So correct answer is B. The DNA is condensed into a chromatin fiber when H1 is associated with nucleosome. So mark B as a correct answer. Question number 4. In a sea urchin DNA, which is double-stranded, 17% of the bases were shown to be cytosine. The percentage of the other 3 bases expected to be present in this DNA are, according to Shargov's rule, amount of adenine is equal to thymine and amount of guanine is equal to cytosine in DNA. यहाँ cytosine 17% है, तो guanine will also be 17% and rest 66% में से 33% adenine होगा and 33% thymine होगा. So correct answer is A. G or guanine is 17%, A or adenine is 33% and T or thymine is 33%. Mark A as a correct answer. Question number 5. The diagram shows an important concept in the genetic implication of DNA. Fill in the blanks A to C. रे डायग्राम देखो, डीएनए से डीएनए का बनना रेप्लिकेशन ऑफ डीएनए कहलाता है. डीएनए से मैसेंजर आरएनए का बनना ट्रांसक्रिप्शन प्रोसेस है. मैसेंजर आरएनए से प्रोटीन का बनना ट्रांसलेशन प्रोसेस है. एंड फ्रांसिस क्रिक described this unidirectional flow of information in 1958 as the central dogma. So correct answer is A. In given ray diagram, A is transcription, B is translation and C is Francis Crick, who proposed central dogma. Mark A as a correct answer. Question 6. What are the structures called that give an appearance as beads on string in the chromosomes when viewed under electron microscope? See the diagram below. These are nucleosomes which appear as beads on string in the chromosomes when viewed under electron microscope. So mark C as a correct answer. Question number 7. Which one of the following does not follow the central dogma of molecular biology? सेंट्रल डॉगमा में डीएनए से मैसेंजर आरएनए एंड मैसेंजर आरएनए से प्रोटीन बनता है, बट इन रेट्रोवायरस लाइक HIV और ह्यूमन इम्यूनोडेफिशिएंसी वायरस, डीएनए इज फॉर्म्ड फ्रॉम आरएनए एंड दिस इज कॉल्ड एज रिवर्स ट्रांसक्रिप्शन. सो आउट ऑफ 4 ऑप्शंस, ऑप्शन डी HIV does not follow the central dogma. Mark D as a correct answer. Question 8. The 3' to 5' phosphodiester linkages inside polynucleotide chain serve to join. Phosphodiester bonds 2 nucleotides के phosphate groups के बीच में बनती है और एक न्यूक्लियोटाइड को दूसरे न्यूक्लियोटाइड से ज्वाइन करती है. सो करेक्ट आंसर इज सी. इनसाइड पॉलीन्यूक्लियोटाइड चेन, one nucleotide join with another nucleotide by 3' to 5' phosphodiester bond. So mark C as a correct answer. Question 9. Which one of the following statements about the particular entity is true? Statement B is true. The gene for producing insulin is present in every body cell. But note one important point about it. This gene is not expressed in all cells, even though it is present in all body cells. Now come to statements A, C and D. These are not true statements. Statement A में देखिए, सेंट्रियोल प्रेजेंट होता है एनिमल सेल में विच प्रोड्यूसेस एस्टर ड्यूरिंग सेल डिवीजन. स्टेटमेंट सी में देखिए, न्यूक्लियोसोम इज फॉर्म्ड ऑफ अ कोर ऑफ 8 हिस्टोन्स. And in statement D, DNA is composed of nucleotides. So, out of 4 statements, true statement is B. Mark B as a correct answer. Question number 10. In the DNA molecule, see the diagram. DNA molecule में there are 2 strands जो anti-parallel run करते हैं. One in 5' to 3' end and other in 3' to 5' end. So, correct option out of 4 given option is B. Mark B as a correct answer. Question 11. One turn of the helix in a B-formed DNA is approximately. Double helical DNA structure में इसका diameter होता है 20 angstroms and one turn has a distance of 34 angstroms which is equal to 3.4 nanometer. इसके अलावा एक और इम्पॉर्टेंट पॉइंट नोट कीजिए, 10 बेस पेयर्स प्रेजेंट होते हैं इन वन टर्न एंड 2 बेस पेयर्स के बीच का डिस्टेंस होता है 0.34 नैनोमीटर. सो करेक्ट आंसर इज डी, 3.4 नैनोमीटर. मार्क डी एज अ करेक्ट आंसर फॉर द गिवन क्वेश्चन. क्वेश्चन नंबर 12. विच वन ऑफ द फॉलोइंग मेक्स यूज ऑफ RNA टेम्पलेट टू सिंथेसाइज DNA? रिट्रोवायरस में RNA टेम्पलेट से DNA का सिंथेसिस होता है एंड फॉर दिस एंजाइम रिवर्स ट्रांसक्रिप्टेज इज यूज्ड. सो करेक्ट आंसर इज सी. रिवर्स ट्रांसक्रिप्टेज uses RNA template to synthesize DNA. Mark C as a correct option. Question number 13. The following ratio is generally constant for a given species. The ratio of G plus C, that is guanine plus cytosine, and A plus T, that is adenine plus thymine, is constant for any species as explained by Chergaff's law. So, correct option is C. G plus C upon A plus T is generally constant for a given species. Mark C as a correct answer. Question 14. Genes are pegged into a bacterial chromosome they are pegged by basic proteins as histones are absent in bacterial cells. So, correct answer is D. Mark D as a correct answer. Question 15. In prokaryotes, the genetic material of prokaryotes is circular and single-stranded DNA. It has no association with histones. So, correct answer is B. In prokaryotes, the genetic material is circular DNA without histones. Mark B as a correct answer. Question number 16. Nucleosome core is made of, correct option is D. Nucleosome core is made up of H2A, H2B, H3 and H4. It is about 7 to 10 nanometer in diameter, having histones around which DNA strand is wrapped. So, mark D as a correct answer. Question 17. Nucleotide arrangement in DNA can be seen by X-ray crystallography. Correct answer is A. X-ray crystallography technique is used to see nucleotide arrangement in DNA. So, mark A as a correct answer. Question 18. The experimental proof for semi-conservative replication of DNA was first shown in 1958. Meselson and Stahl proved that replication of DNA is semi-conservative by using bacteria Escherichia coli or E. coli. So, correct answer is B, B. Question 19. The final proof for DNA as genetic material came from the experiments of Hershey and Chase in 1952, worked with viruses that infect bacteria and gave final proof that DNA is a genetic material. So, correct answer is A, mark A. Question 20. Taylor conducted the experiments to prove semi-conservative mode of chromosome replication on. After Meselson and Stahl, Taylor and colleagues in 1958 performed experiments on Vicia faba, also called faba beans, and proved the semi-conservative mode of chromosome replication. So, option B, Vicia faba is correct answer, mark B. Question 21. Transformation was discovered by. In 1928, Griffith did series of experiments with Streptococcus pneumoniae bacteria and witnessed transformation in bacteria. So, correct answer is C. Transformation was discovered by Griffith. So, mark C as a correct answer. Question number 22. A molecule that can act as a genetic material must fulfill the traits given below except. Trait A देखिए, it should be able to express itself in the form of Mendelian characters. This is correct for a molecule which act as a genetic material. Second, it should be able to generate its replica. Yes, this is also correct trait of genetic material. C, it should be unstable structurally and chemically. No, this is not correct. The molecule that can act as genetic material should be stable structurally as well as chemically. Now come to D. It should provide the scope for slow changes that are required for evolution. This is also correct trait for a molecule which act as a genetic material. So, answer is C. Mark C as a correct answer for the given question. Question 23. The unequivocal proof of DNA as genetic material came from the studies on a. This study was done on bacterial virus that is called bacteriophage or virus which attack bacteria. So, answer is D. Mark D. Question 24. The transforming principle of pneumococcus as found by Avery, MacLeod and McCarthy was. Work of Avery, MacLeod and McCarthy proved that it was DNA and not proteins or RNA which could transform pneumococcus bacteria and concluded that DNA is the hereditary material. So, mark A, DNA as a correct answer. Question 25. Which of the following hydrolyzes internal phosphodiester bonds in polynucleotide chain? The enzyme endonuclease hydrolyze the internal phosphodiester bonds in polynucleotide chain. So, correct answer is B. Mark B. Question number 26. Escherichia coli fully labeled with N15 is allowed to grow in N14 medium. The two strands of DNA molecule of the first generation bacteria have. जब fully labeled with N15 E. coli को N14 मीडियम में ग्रो किया गया, then फर्स्ट जनरेशन में जो 2 DNA के स्ट्रैंड्स मिले उनकी डेंसिटी पेरेंटल DNA से डिफरेंट मिली इन द एक्सपेरिमेंट. So, correct option is A. the two strands of DNA molecule of the first generation bacteria have different density and do not resemble parent DNA. So, mark A as a correct answer. Question 27. In a DNA, percentage of thymine is 20%. Then, what will be the percentage of guanine? See the solution. If thymine is 20%, then adenine will also be equal to 20% as both are equal, and A plus T होगा 40%. After this, guanine plus cytosine का amount होगा 60%, and as guanine is equal to cytosine, so guanine will be equal to 30%. Correct answer is B. Guanine is 30%. Mark B as a correct answer. Question 28. DNA synthesis can be superficially measured by estimating the incorporation of radio-labeled. DNA synthesis को measure करने के लिए radio-labeled thymidine को use किया जाता है because thymidine is not there in RNA and RNA will not be labeled by this. So, correct option is A. Mark A. Question 29. Similarity in DNA and RNA is that option A देखिए DNA and RNA दोनों ही polymer of nucleotides हैं. Both are polymers of nucleotide. B option. DNA में pyrimidine thymine base है while in RNA it is uracil. So both have different pyrimidine. C option देखिए DNA has deoxyribose sugar while RNA has ribose sugar, thus both have different sugar. D option. DNA is considered as genetic material and not RNA. So statements B, C and D correct option नहीं है, while statement A is correct answer for the given question. Mark A. Question number 30. Which one of the following is not applicable to RNA? Chergaff's rule is not applicable to RNA, while RNA has heterocyclic nitrogenous base. Sometimes it also has complementary base pairing and it has 5' phosphoryl and 3' hydroxyl ends also. So, answer is B. Mark B as a correct answer. That's all for today. Thank you very much for staying with me. Subscribe to the channel to get the information about my new video. Thank you so much.